Hello, my name is Eleanor Warren, and today I'll be speaking about the presence of CHK1 in the nucleus of transfected U2OS cells in relation to sites of DNA replication.
Where did you get the idea for your project, experiment design, and analysis?
So my project was inspired by my PI and a paper that they had published previously. And the main issue with the findings that they had were that their fluorophores and their fluorescent proteins connected to the CHK1 were too bright and overexpressed, causing a phenomenon known as spectral bleed-through. And the issue with spectral bleed-through is that it doesn't really give you conclusive results. So my project was to focus on using dimmer fluorescent proteins to view CHK1 in the nucleus and see where it localized.
What interests you about this topic?
So I think my interest mainly stems from biology. When I first got there, I didn't really even have a good baseline knowledge of what I was going to be doing, and I guess slowly as I started understanding what I was doing, that kind of made it more interesting. So I think that that's ultimately what got me into doing this project.
What question are you trying to answer, and how did you decide to go about answering it?
So the question I tried to answer was, where does this CHK1 that's tagged with two different fluorescent proteins localize in the nucleus in relation to where DNA is replicating? Since CHK1 is in an active DNA kinase, it should be present at sites of DNA replication.
What did you learn about the science behind your project before and during the experiment?
So I think the main thing that I learned about the science was that my concept of science, before this happened, was kind of the idea of being in a science class where you really know what your ending answer is, where you know that if you end up with a mistake in your answer, it's somewhere in your procedure. And ultimately that's not the case in the real world. In the lab that I was working in, we may not have known if it was the hypothesis or if it was the procedure or bad reagents or whatever. We don't know if that could have been what would have caused a bad result. So I think also one of the main things I learned was that really in reality scientists fail 90% of the time. But that 10% that they get their answers and they get the answers that they want make it all worth it. So I think that that's at the end of the program, at the end of the summer, that's what I had to come to terms with I was doing science. I had a lot of struggles, but I got a lot of reward from the answers that I found too.
What did you learn from the data?
So really what I learned was that in reality, in what I had found, CHK1 did not localize where we thought it would. So the main way that we viewed the CHK1 was that we took a plasmid that has shadow Y and M turquoise on it, right next to the CHK1, and we transfected it into U2OS cells using a lipofectamine 2000 protocol. And really the goal with this was to get the CHK1 to overexpress and make it visible to fluorescence resonance energy transmission, otherwise known as FRET. And really when viewing FRET, there's some things you kind of have to keep in mind. So the best analogy I can give you is if I use my hand. So imagine that there's shadow Y on this end and there's M turquoise on this end. Shadow Y is a yellow fluorescent pigment, and turquoise is a turquoise fluorescent pigment. And when FRET is off, they're separate. There's more than 10 nanometers in between them, and there's active CHK1 since it has an open catalytic site. And when there's FRET on, the fluorophores are excited, so they come within several nanometers of each other, which there isn't a visible discernible difference on a fluorescence microscope. And this closes off the CHK1 and makes it inactive. So this allows us to view where active CHK1 is and where inactive CHK1 is, which is pretty important. In terms of where it is with the DNA replication, we use something called an EDU staining protocol. EDU itself stains for rapidly proliferating DNA, which is U2OS, since it is the cancer cell line. So EDU stains for active DNA replication, and you can see here with the purple by itself. And the EDU binary is otherwise known as EDU positive. So where there's red, you can see in between there's some blue and yellow. And this is a little bit concerning because remember that the blue and yellow mean that there is active CHK1. So where there was active CHK1, there was no DNA replication occurring, which was odd to us because CHK1 is an essential DNA kinase, so in theory it should be at sites of DNA replication. And you can see additionally confirmed here that there is green in between EDU non-binary, so active CHK1. so inactive CHK1. So these results were a little bit concerning to us, so we looked at the spectral bleed through, make sure we weren't making a mistake. And the spectral bleed through showed that there wasn't really a huge issue. So the main problem with FRET is spectral bleed through, where the acceptor fluorophore is overexpressed, and it kind of autofluoresces the other colors, so you don't necessarily know which is true. And in this case, the shadow Y is an acceptor fluorophore, and the M turquoise is the donor fluorophore. So you can see here that there's not really a significant amount of spectral bleed through. So we looked back at the DNA I was using. And between two different clones of the same DNA, we found that there was a line of a plasmid that popped out that wasn't really, it wasn't supposed to be there. So this could be degraded DNA, it could be a contamination, whatever, it was not good. We also looked at this through Western blotting and immunoblotting. You can see a little bit of the technique here, but the Western blot results that we got were that there was a double band in the shadow Y CHK1 M-Turk compared to the M-Cherry CHK1 M-Neon. So the M-Cherry CHK1 M-Neon was a previously published plasmid that's confirmed to have the CHK1 in it. We confirmed the presence of CHK1 with our Western blot in my plasmid. And so this double band could mean multiple things. It could mean degraded proteins. It could mean a contamination that had the same signal for the same antibodies that it was blocked in. And you can see that there's also some innate CHK1 expression in all of them because CHK1 is an essential DNA kinase. So really what I learned from all of this was that CHK1 localized over non-selective DNA replication, but the main way that I would do this in the future, or what I would change, is that I would do the protocol multiple times because I believe that this is an outlier result. And I could also change the transfection protocols to increase the transfection efficiency, so how many of the cells actually pick up the DNA. And I believe that that is it. I would love to thank my program at UPMC, the Hillman Academy, for letting me have this experience.